Hi everyone, welcome to the first video of Chapter 5. This is the beginning of your Exam 2 material, and we're starting with Section 5.1 from your textbook. This is going to give you an introduction to the study of tissues. In general, for Chapter 5, we're going to be talking about histology. Histology is looking at cells, classifying cells, and looking for abnormalities in cells. So here we'll be looking at the four broad tissues: epithelial, connective, nervous, and muscular, and looking at the cellular structure of those tissues. Please keep in mind what an organ is. An organ is a structure that has a discrete boundary. You can tell what is part of the organ and what is not part of the organ. And the organ has to be made of two or more tissue types in order to have that classification. Also, histology, we've talked about this definition before. Histology is the study of tissues. Here in section 5.1, we'll be talking about those four classes of tissues and what makes things fall into one class versus another. I want to give you a very brief introduction to embryonic germ layers and how the fertilized egg starts to differentiate and then continues differentiating all the way until we have all of the adult tissues that we'll be studying. And then we'll talk a little bit about how to figure out what a three-dimensional structure looked like based on two-dimensional tissue sections. So, a little bit of a review. A tissue is a group of similar cells that all work together to help an organ function. Like we said, there are four different types of tissues. We have epithelial tissues, connective tissues, nervous tissues, and muscular tissues. And there's a lot of variation within each of these categories, and we'll look at each one of these categories in depth. They differ from each other in what types of cells they have and how those cells work together. They also have different extracellular matrix, or the material that is surrounding them. And they are also different in the amount of space that is taken up by cells versus the matrix that surrounds them. So let's take a closer look at this extracellular matrix for just a second. The extracellular material, or extracellular matrix, is the thing that surrounds the cells. It's generally composed of two different things. The first is proteins that are kind of long, stretchy, fibrous proteins. And between those proteins, a clear gel. And that clear gel can also be called tissue fluid or extracellular fluid or interstitial fluid or tissue gel. My favorite term, the one that I'll use most often, is extracellular fluid, but there are several synonyms for that for you to be aware of. So we know that development begins when a sperm cell fertilizes an egg cell, and we have two sets of chromosomes at that point. And almost immediately, that fertilized egg, which we call a zygote at that early stage, starts to develop by cell division. This happens by mitosis that we've talked about previously. So without getting too deep into developmental biology, that's its own very fascinating field. But very briefly, as the fertilized egg starts to develop in the very, very early stages, one of the first things that happens is we see three layers start to form. And these three layers start to differentiate and eventually develop into specific cell types. Okay, so we have the ectoderm. Okay, so ecto generally means outside. When you see ecto as a prefix, think outside. Okay, so the ectoderm is the thing that's going to develop into the epidermis and the nervous system. Okay, so we can see the ectoderm in this this diagram. We're looking at the green here. Okay, so the ectoderm starts to develop as the cells divide by mitosis. Then we have the endoderm. Whenever you hear endo, think inside or inner. Okay, so the endoderm is the yellowish, orangish tissue here, and it's starting here, the endoderm. All right. And the endoderm is going to develop into the digestive tract, the respiratory tract, also the liver and the pancreas. And then the mesoderm, and sometimes people might pronounce it mesoderm. I believe both are correct. I've heard experts say it both ways. This mesoderm is the middle layer. Okay, so we can see here in the diagram, we have the purple section, and this mesoderm is developing in between the green ectoderm and the yellowish-orange endoderm. That's our mesoderm. And it's going to develop and differentiate into the muscles and the skeleton. Additionally, blood and cartilage are going to come from the mesoderm. Okay, so now very briefly, how do we get the slides that we're going to be looking at through the rest of this chapter and really all of anatomy and physiology? We need to understand how slides are prepared. How do we get the sections that we see? Okay, so first of all, cells are generally fixed in formalin. It prevents decay of the samples and kind of holds everything in place exactly as it is at the moment that it's fixed. What we call sections that are put on slides to be viewed with a microscope are super, super thin slices. They're usually only 1 or 2 cells thick, and that allows us to see through the less dense portions of the cells with a light microscope. So the way they get these sections is using something that looks like a miniature deli slicer. Like if you've ever been at the deli and you're gonna get a pound of turkey or a pound of ham, and they use the rotary slicer, and it slices thin slices of the ham or the turkey. A miniature version of that is what's used to make these super, super thin slices of cells. Then we choose the stain that we want to add to the slides depending on what we're interested in seeing. We know that some cell components will look different colors or will take up stains differently. And so we choose the stains that we use based on what we're hoping to observe. It's important to remember that these slices, these slides that we look at, are always a 2-dimensional representation of a 3-dimensional structure. So talking just a bit more about that, I like this macaroni example up here. So if we're looking at a plain old macaroni noodle and we were to cut it in 3 different places, we would see a very different picture of that macaroni noodle. If I were to cut it where this red line is, I would see 2 hollow circles. I would have no idea that those hollow circles were ever connected. If I were to slice where this green is, it kind of appears I have a bowl shape and a bowl shape that are connected. And if I were to slice where this blue line is, I would just see an oval-shaped bowl. Now, putting all 3 of these together, I might be able to interpret that they came from a macaroni. Okay, this is not a human example. This is called an amphioxus. It's a little sea creature that I have done some work with in other classes. So an amphioxus is this kind of long, narrow, kind of looks like a tadpole creature. And depending on where we take a cross section, cutting straight across, we see very different structures on the inside of it. So in this one, this is from the front of the body, and we can see this hollow area kind of where its mouth and throat structures are. If we take a cross section around here, we see this big hollow opening. If we move back, we can see some of the reproductive structures. If we take a cross section here and a cross section here at the end, we're seeing mostly muscles because that part of the body is used for motion, for moving the tail. So taking cross sections at different places give us a very different view of that animal. And I'm sure you guys can imagine if you were to take cross sections of human structures at different places, we could also get very different pictures of what we're seeing. So a few more vocabulary words that are going to be important for you. The position that we look at the specimen from really changes our view. Okay, so we can look at a longitudinal section. A longitudinal means it's along the long axis. So if we're looking at a long bone, this could be a femur, could be a humerus here. If we were to look at a cut through the long axis, we would see kind of this shape of the whole bone. If we were to take a cross section and cut straight across, we would see the outside and the inside of the bone. And an oblique section is something that's not longitudinal and not a cross section. It's kind of an intermediate between them, and we would see more of an oblong shape. So we have to be very aware of how a slide was prepared in order to interpret what we're seeing.